Yeah, so thank you. So I'm here today to present the DigiBrain project. So I'm really happy. So first of all, I'm very happy that you were able to organize this event, although in these very difficult circumstances. So our project is a quite large one. It consists of 9 partners. I will not be presenting results from all of it, but I'll try to walk you through one of the examples from the project, which may also illustrate some of the challenges that we have met during the project. And as you may already know, of course, the brain-related diseases are among the largest health sciences or health challenges that we have in the world. We expect that at least now 1/3 of the population may be affected, and it's expected that this will also increase in the years to come when we have an aging population. The diseases that we are focusing on in this project are mental illnesses, and these are called clinical syndromes, and that means that we don't have any known biomarkers. So it's the diagnosis is then set by the psychiatrist in conversation with the patient, looking at also the history of the patient. But the problem then is that we have very inefficient treatments, and the ones that we do have, they may have severe side effects. So, but still, if you look at the heritability, it's a high heritability of these diseases. Here you see the schizophrenia quite on top compared to, you could look at diabetes, which was presented here before. It has much lower heritability. If you look at about twin studies. And this must mean that there are some biological mechanisms underlying these diseases. And with the more or the cheaper and cheaper or the ways that we could sequence today, the cheaper or the sequence is much cheaper today than it was before. That means that we could sequence a lot of patients and controls and compare what kind of gene variants are common to these patients compared to controls. And one of these consortia is Ole Andreasen is part of that. And you see here on the right that they have sequenced in 65,000 schizophrenic patients and compare that with more than 200,000 healthy controls. And what they then find is that there are about 270 gene variants associated with schizophrenia. This tells you that, of course, this is a very complex and polygenic disease, but that there may be some genetic causes underlying at least the onset and also the symptoms of the disease. So, and many of these the gene variants are associated with neuron function. It's illustrated in this cartoon where you have the letters illustrating the different genes in a neuron, which is kind of this round cycle. And we have, so you could ask them, where should you actually begin to explore this? We have focused on genes that are associated with ion channels and that are involved in synaptic, like the neural communication in the synapses. And why we do this is that the synaptic dysfunction is actually a hallmark of many of these mental illnesses, likely because they are then the neural communication related to cognition and emotion and behaviors. But still there is a lot of them, so then how should you actually approach this? And what we have done there is that we try to link the gene variants to the clinical symptoms with a more multi or interdisciplinary approach. We try to make computational models at each level, and then have targeted animal and other kinds of experiments to test the predictions from these models and also to go more into the detailed background mechanisms. So then of course you could say that while we work with fish and mice, you can't create a schizophrenic fish or a schizophrenic mouse. So that means that we need to find some basic principles to investigate, which we don't need to ask the fish or the mouse what they feel or what they feel. So some robust findings, and I have kind of this is like we'll come back to that, but at least some findings from the literature is that in schizophrenic patients you see that there's increased cortical excitability and impaired pre-post inhibition. I'll explain that in a moment. So that was kind of the basis for the project. And to just first to explain what this pre-post inhibition is that you could think of like if you have a very sharp sound, you'll startle. All animals will do that. But then if you have a weaker sound that precedes this very sharp sound, both we and all other animals will actually have a less of a response to that sharp stimulation. And then with the patients with schizophrenia, they show an impaired startle or this pre-pulse inhibition, so that they have the strong response even though they have this pre-pulse, and it's thought that this is because of an increased neuron excitability. So to explore this, then we started out to make computational models of each level to try to have a more high throughput exploration of the effects of these gene variants. So Tumo and his colleagues then made different neural models, and then he looked at what was the effect of the known gene variants to these neuron functions. And what he found was that there were very, very little or small effects if you looked at each gene variant. But if you combine them, then actually it had a substantial effect on the neural function. And it also showed that it actually did lead to a neural excitability, so increased neural excitability, which then was similar to what was reported in the literature for schizophrenia patients. But then this was a single neuron, but in order to look at what is the network effect, what was the effect on the EEG signal of the patients, what we then did was that instead of having one neuron, we actually combined them. So 100,000 more biophysically detailed neurons, combine them into a network, and then try to extract then the population activity, the EEG activity. And by using this model, then Tumo then could then put again his gene variants into that model, and then what he found was that he could kind of reproduce what was then reported in patients, that there were some disturbances in some of the frequencies in the EEG signal. So of course this seems to be that there's some resemblance or that he could reproduce some of the findings from the clinic. But then how would this relate to the animals? So for that, then Nancy, she now uses zebrafish larvae, and she uses the very same paradigm as you use in humans for pre-pulse inhibition. So she presents a very sharp sound to these small larvae and then precedes that with a weaker stimulation. And what she found was that if she looked at mutants that had a mutation in one of the very severe or one of the risk genes that we find that the most severe schizophrenia patients also exhibit, then she showed that actually it's a reduced pre-pulse inhibition in this larvae. And then when she put the electrodes into the head of this larvae, she also could show that there is some kind of increased neuron excitability. And there, of course, this is one way to look at neuron excitability. There are other ways to induce brain excitability. One way is to increase excitable neurons, but you could also decrease the dampening effect of other kinds of neurons, so you could decrease inhibition. And we did that in mice, and we showed them in the now again this pre-pulse inhibition paradigm that the mice startled more if they had a reduced inhibition. So if the brain was more excitable, it was an increased startle effect. But what we couldn't show, we couldn't find that there was any difference in the pre-pulse inhibition, even though they were startling more. So of course, these were only then animal studies, but how does that relate to our own recordings from humans? And then Torbjørn Elsåshagen with his team at Oslo University Hospital, they have now did recordings of quite many patients and also hundreds of controls in very similar paradigms, so these pre-pulse inhibition paradigms and some others. And then what they found now is that actually in their recordings, they don't find the things that are established in the literature. So they do find some indication that there are an increased startle response, but they can't find an impaired pre-pulse inhibition, which is very much established in the literature. They also find that there are no, if they test directly, they can't find any increases in the cortical excitability. They do find, which may be expected, I don't talk about this now, is a decreased cortical plasticity. So this has kind of been, it was quite surprising for us because this was not what we expected. And we don't know the causes of this. We may speculate that there may be some difference in the medical treatment history of the patients that we now are investigating compared to the ones that are in the literature, but that's not known. It may be from different subgroups of the disease. But what now Torbjørn and his team are doing is that they try now to match the genotypic profiles that they do have of these patients with the EEG measurements, and we also do phenotyping of patient-derived pluripotent stem cells to try to see if there are any links. But this then leads me to, at least I was asked by the Digital Life to also to address some other questions. And that was, what if we had started today with the project? And I think that one of the things that we would have done, or may have done, was that we would have liked to have more in-house proof of concepts from the beginning. So we based kind of these findings from the pre-pulse inhibition on literature and not our own findings. And we based many of these experimental approaches on that notion. So we would have done that differently. And I think that overall, at least this kind of weakness of the project as we now see it, or at least as I see it, is that perhaps we should also use less model system and less approaches, because many of these methods have been established within the project, which has been quite time consuming, and then the results are then perhaps also delayed. But then again, our aim of the project was actually to establish this platform or this approach, so that could defend it, but perhaps also it may have been easier for us to apply this approach on a disease which has a less polygenic complexity. That's kind of what we see now.
1 minute, Marianne, 1 minute.
Yeah. And then also we were asked to talk a little bit about the experiences from being part of the Digital Life Center. And I think it's has been very rewarding to be part, or I feel that we have been part of an open-minded organization and been able to contribute to the development. We have had some very nice arenas which we wouldn't have else, or to talk about, like these research, the responsible research and innovation. We also have many opportunities for support, and but there sometimes has been difficult to understand the expectations from what we should contribute with into the center. And yeah, so I think that I'll just stop there. I think that this integrated and interdisciplinary approach that we have tried to establish is actually, it has a future to address challenges or questions for disease. And it seems to be that some of these neural models could reproduce some of the findings in the clinic. And also that we could, if we are able to identify basic traits of the disease, we could also then investigate those more into detail.